Hello there, and welcome to Fundamentals of Biology, Lesson 3, where we're going to be talking about carbohydrates. So this follows on from the last session then, where we looked at elements and atomic structure, and then we joined atoms together to make ionic and covalent bonds. And today we're going to take the next step from that and start to look at organic compounds. So the objectives for this session then are to have a look at organic compounds, and in particular the first of four classes of organic compounds, which are going to be the carbohydrates. And then we're going to have a look at how we actually go from the simple carbohydrates up to the complex carbohydrates, what we would class as macromolecules, so large molecules. And we do that through a process called dehydration synthesis. And then we'll also look at how we break those macromolecules down into their constituent parts again through a process called hydrolysis. So that's the plan of attack for this session. So before we begin, just to quickly recap on where we finished in the last session then, and I ended by telling you that there are four main classes of organic compounds, and they are carbohydrates, lipids, proteins and nucleic acids. So today we're going to have a look at carbohydrates, and then in the next session we'll look at lipids and nucleic acids, and then we'll finish this section off by having a look at proteins. And all the way through you'll see that there are quite a few similarities, particularly in the processes that are used to make and break the larger molecules. So let's get on and we'll have a look. As we go through these next few sessions then, there's going to be a couple of terms that I use frequently, and they are monomer and polymer. And just to have a look at this diagram, we can see that monomers are represented by those single sort of orbs there. And that's basically what monomers are. They are single units of something. So in this case, they're represented by these sort of green shapes. And each one of those would be an individual monomer. If we start joining those together, as you can see at the bottom there, we go through a process called polymerization. And this is the joining together of the individual monomers. And at the bottom there, you can see that they are now forming a long chain. And we would refer to the long chain as a polymer. So it's a bit like taking a bead necklace. Each individual bead would be a monomer, and you join them all together to make the necklace, and the necklace would be a polymer. And we're going to see this happening in carbohydrates, in our nucleic acids, and in proteins. So polymers in these cases would be referred to as the macromolecules, these really large molecules. And the monomers will be the individual building blocks that make them up. But notice I didn't mention anything about lipids then, because lipids are the odd one out of our four classes, in the sense that lipids do not tend to form these large macromolecules. And we'll look at that in more detail in the next session. But keep in mind that monomers and polymers, those are terms that I'll be using quite a lot. And the carbohydrates, nucleic acids and proteins each have their own specific monomers, and then they will also have their own specific polymers. So there's going to be quite a few words like this that I introduce you to during these sessions. So my recommendation would be to, if anything comes up that you're not sure of, is to make a note of it, just jot it down, and then you can always look it up later, because one of the hard things about learning biology is the sheer amount of new words and terminology that comes your way, and it really does help if you can get on top of it. Let's start having a look at carbohydrates then. So what are they, and why are they essential for life? Well, first and foremost, if I asked you to tell me what is the first word is that springs to mind when you hear the word carbohydrate, it's probably something along the lines of energy or food or bread or potatoes or pasta or something like that. It's going to refer to that food and energy that we know our bodies require. And that's no accident. So carbohydrates are the principal and preferred source of fuel for the body. So our bodies require food to produce a currency of energy that our bodies can actually use, and that's called ATP. adenosine triphosphate, and we'll look at that in a later lesson. But our bodies use the molecules in our food to actually produce that. And the food that they actually prefer are carbohydrates. So it's a bit like putting petrol in your car. If you don't put petrol in the car, you're not going to get very far. And it's a bit like that with our bodies. We need to keep putting food in them to keep generating the energy we need to do all of the things that we do. So out of the carbohydrates, and as we'll look at as we go through this session, they're a very diverse group. We'll see that glucose, which is a simple sugar, so one of the smallest carbohydrates or sugars that we come across, this is the principal circulating sugar in the blood, and it's also found in most plants and other animals. So it's not just us that uses this as a fuel source. Most forms of life that have access to glucose do tend to prefer it as a fuel source to produce their energy. So we'll talk more about that as we go through. And as we work through the module, we'll see that we can actually use other fuel sources as well. The body is very adaptable, and we're able to use fats and proteins as well. But given the choice, the body does tend to prefer glucose. So why do we need it? Why do we need this fuel source that produces energy? Well, it does an awful lot for us. So not only can we use the carbon within the glucose molecule or within our carbohydrates to actually become carbon skeletons of other molecules that physically build our bodies, we're using them to produce that molecule ATP that I mentioned just now. And that gives energy to the body for all of the functions that it does, really. So proper muscle function, organ function, and also for our brains to work properly. And our brains are actually incredibly hungry organs for glucose and for that energy that it produces. So it is it's very, very important for the healthy functioning of a body. But this is an important thing, an important point. So although we tend to think of carbohydrates as a fuel source, and we think of food when we hear the word carbohydrate, it's a really diverse group of molecules, and they don't just come in the form of fuel. They actually have a lot of other functions, a lot of other roles. So as we'll look at as we work through this session, we can also get really large macromolecules in our carbohydrates, and they can actually become storage mediums, so they can store energy for later use, but they can also make physical structures, and we refer to those as structural carbohydrates. And classic examples are the exoskeletons of arthropods. So arthropods are those animals that have exoskeletons and jointed limbs. So we're looking at things like insects and crustaceans and arachnids as well. And that carbohydrate is called chitin. So you can see it in brackets there. It's not chitin. It looks like you could pronounce it that. It's actually pronounced chitin, but you pronounce it any way you like. So the exoskeletons that we find on crabs and lobsters contains, there's some protein in there as well, but it's largely made up of this carbohydrate called chitin, which gives it strength and flexibility. But we also find it making another structure, and that's cellulose, which we find in plants. And cellulose is the most abundant organic compound on the planet. It makes up cell walls and of all plant cells. It's also an important component in bark and wood. So it enables trees and plants to stay upright, gives them strength and rigidity. So it's an incredibly versatile and important group of macromolecules. And as you can see there, also we can get starch in plants, which is a storage carbohydrate. So it's a store of energy for later use. So most of our carbohydrates then enter into food webs by photosynthesizing plants. So plants use the energy of the sun or light and carbon dioxide and water to photosynthesize and produce simple sugars like glucose that we mentioned just now. And from that, we get our carbohydrates entering into the food web. So what are the implications for carnivores, which I've written there? Well, if you're a carnivore and you don't eat plants, so you're not able to get your carbohydrates directly from the plants, where are you going to get them from? Well, the first thing that you do is you would try and eat an animal that eats plants. And that's why when we look at the animal kingdom, typically, not always, there tends to always be exceptions to every rule in science or in biology, definitely. But typically, we will find that most carnivores have a preference for eating herbivores. And if you go onto the African plain, you typically don't find lions hunting and killing other carnivores like cheetahs or even hyenas, things like that. They might kill them in a dispute, but they typically aren't actively hunting them for food. Instead, what they're doing is they're hunting herbivores, so zebra and wildebeest and gazelle, and that's because they're closer to this source of carbohydrates, and also the other vitamins and minerals that you get within the plants. And one of the things that you often don't see on documentaries, like David Attenborough documentaries, for instance, is when large carnivores take down a herbivore, like for instance on the African plains, you don't see often what they do once they've killed that animal. The first thing that they'll tend to do is they will actually go into the animal through the stomach, through the abdominal cavity, where they'll then sort of eat the intestines and the stomach contents, and then they'll also eat the organ meat. And once that's done, they'll then move on to the larger muscle meat. But they'll eat the intestines and that because they will contain the partially digested carbohydrates that they can then hopefully access. And we'll see why they do that and why most animals aren't able to digest plants, or particularly the cellulose in plants, in a few slides' time. So carbohydrates then, very, very important source of fuel for the body, but also can make structures and storage media as well in plants and animals. So what actually are carbohydrates then in terms of their sugar and in terms of their structure, and where do we find them? Well, carbohydrates include both sugars. So as we'll see when we go through, sugars we tend to refer to as the smaller carbohydrates, what we actually call mono and disaccharides. But we also get them carbohydrates in the form of polymers of sugars. So if we take something like glucose, which is a small carbohydrate, what we refer to as a monosaccharide, if we join lots and lots of glucoses together, so join lots and lots of monomers together, we can actually make a polymer, or what we refer to as a polysaccharide, and we get things then like starch and cellulose. So we get not just the sugars, we also get the polymers of sugars. So the simplest sugars then are the monosaccharides. So mono meaning one, saccharide referring to sugar. And we often refer to these as simple sugars. So these are just individual sugars, and we'll look at the structure of these in a minute. If we join two of these monosaccharides together, we get a disaccharide. So disaccharides are double sugars consisting of two monosaccharides joined together by something called a dehydration reaction, or through dehydration synthesis. Now, depending on where you've learned biology before, if you have, you might have heard of these reactions referred to as condensation reactions. So condensation or dehydration reactions both mean pretty much the same thing in this context. So either word is absolutely fine to use. So if you've learned this process as condensation before, you can just keep going with that. If you say that to another biologist, they'll know exactly what you mean. So it's not a problem. So we have monosaccharides, which are simple or single sugars. We join two of those together, we get disaccharides, and I'll give you examples of these in a second. The next stage we can get is once we start getting to 3 to 10 simple sugars joined together, each one joined onto the others through dehydration, we get a group called the oligosaccharides. These aren't particularly common in terms of, we don't talk about them a lot, but they do have some important roles. So they form important tags on the outside of our cells, for instance, that enable cells to recognize each other and to recognize pathogens, for instance. So they sort of enable this cell-cell recognition. But we don't talk about them a huge amount, but I'll give you some examples when we talk about cells in the future. So oligosaccharides then are formed when 3 to 10 simple sugars are linked together. And then we get the really big ones. So what you'll often hear referred to as complex carbohydrates. And these are what properly called polysaccharides. So poly meaning many, saccharides meaning sugar, so many sugars. And polysaccharides are polymers composed of many sugar building blocks. So for instance, the examples that we're going to look at in a minute, they're all composed of hundreds or thousands of individual glucoses all joined together. So where do we tend to find these then in our everyday life? Well, I've got some examples there on the little diagrams at the bottom. So on the left, we've got examples of simple carbohydrates then. So with simple carbohydrates, we're referring to the monosaccharides like glucose, but also a lot of disaccharides, things like sucrose or normal table sugar. So we tend to find these in abundance in a lot of our favourite foods, unfortunately. Things like sweets and biscuits and chocolate and cake and fizzy drinks, but also in some healthy things like fruit. And we're often told, and we often hear that sugar is becoming a really big problem in the modern diet. And there's a good reason for this, and I'll try my best to explain it. So if we go back thousands of years into our evolutionary past, we were drawn to sweet foods. And if you go back 5,000-6,000 years ago or even longer, the only real place that we could find sweet foods were in the form of fruits and berries and some honey. And we were drawn to these foods, particularly the fruits, because not only are they full of these sugars that were hard to come by, typically throughout the year, so when you could get them, they were a really, really good source of energy that was important then. But they also came packed with lots of vitamins and minerals that also were beneficial to the diet and to the body. So we are hardwired through evolution to seek out and sort of devour foods that contain these simple sugars, because our brains tie them in with other things that are healthy for us. But we've kind of hijacked that sort of hardwired brain that we have, to basically take our love of these very, very sweet foods and produce them en masse into very sort of cheap and easily accessible foods now. So we're still attracted to the sweet stuff. So we really like cake and sweets and fizzy drinks, but all we're really getting from those foods these days are just the sugars. They don't come with any of the vitamins and minerals or fiber that will actually benefit our diet. They're just packed full of sugar to the point where it's becoming a bit of a problem throughout the world. The other thing to mention before I move on from that is, again, if you go back 5,000-10,000 years ago, these foods tended to only be available seasonally. So, fruit tends to grow at different times of the year. You get some in the spring, summer, and some in the autumn, like apples. But then you'd go through quite a long period of the year where these weren't available to you. Whereas now that isn't a problem. We can go into any shop, pretty much anywhere in the world, and we can fill our pockets with all of the sugar that we want for a pretty low price. So we have access to as much sugar as we want, 24 hours a day, 365 days a year, and we don't necessarily have the willpower to sort of not consume it, and that's where the problem comes. So on the right there, we can see the complex carbohydrates, or what we'd refer to as the polysaccharides, and we tend to find these in we can see it there. We can find it in wholemeal breads, so whole wheat breads, legumes and wholemeal rice and wholemeal pasta, but also in starchy vegetables, so potatoes and sweet potatoes. And the reason that these are sort of better for us is as long as they're not overly processed. So if you can go for wholemeal rice and wholemeal pasta, they tend to come with a lot more vitamins and minerals than the heavily processed white bread, white rice, white pasta. And the other important component of this is they tend to release their energy much more slowly than the simple carbohydrates. So the way I tend to look at this is, if you think of your metabolism as a furnace, as a fire, and you need to keep putting fuel on that fire to keep producing energy. Well, the simple carbohydrates like glucose and sucrose, they're very easy for the body to process. They're very easy for the body to digest and then move into your blood and then move into your cells where they can start getting turned into energy. And I would equate this to sort of pouring petrol on a fire. I don't recommend that. That can be very dangerous, by the way. But it's like putting petrol on a fire. You get this really big, explosive, rapid burning of this fuel, and then it goes very, very quickly. And if you consume a decent amount of sugar, you probably would have recognized this yourself. If you wake up first thing in the morning and you're running late, and for breakfast you have a big slab of cake and you wash it down with a can of Coke, you will probably run out the door fired right up with energy. And then about an hour later, you'll start getting what's referred to as a sugar crash. You'll start feeling headachy and a bit jittery, and you might even get a little bit hangry. Your temper starts to get a little bit frayed, and that's because you've had this rapid release of energy, and then it's just gone. It's burnt very, very quickly. With the complex carbohydrates, or the polysaccharides, so if you wake up in the morning and you have oatmeal, so porridge, for breakfast instead, what you'll find is, because those carbohydrates are locked up in these much larger molecules, the body it takes longer for them to digest and to process. So you get this much more gradual release of energy. So although you might not receive the same sugar high that you would get from the cake and the can of Coke that you had for breakfast, instead you'd get a much longer lasting sort of full feeling, and you would stay energized for longer, and you should avoid the sugar crash. And it, on a more biological note, it can really help to moderate your blood glucose level. So it's not sort of rising and falling or booming and busting throughout the day. So the complex carbohydrates then are a much healthier option, not just from gradual energy release, but typically they will come with more vitamins and minerals and fiber that also benefit the body in other ways. Okay, so that's sort of dietary advice over. We can get back to the lesson. Okay, so now we know a little bit about what carbohydrates are. Let's have a look at their structure. Carbohydrates come in a range of forms then, with a differing number of carbons. So if you remember at the end of the last session, I said that the study of organic chemistry is the study of compounds and molecules that contain carbons, and carbons are the main feature of carbohydrates. So their general formula can be given as some form of CH2O. So I don't want you to be put off by that. It's fairly straightforward to follow, particularly when we're talking about the monosaccharides and some of the disaccharides. What this means is there will always be, or typically there will be, twice as many hydrogens as there are carbons and oxygens. So in this instance, we've got CH2O, but if we were to have a 3-carbon carbohydrate, that would have the formula of C3H6O3. So 3 carbons, 6 hydrogens, and 3 oxygens. So we can see there are twice as many hydrogens as there are carbons and oxygens. And this is an actual carbohydrate that exists. It's called glyceraldehyde, one of the precursors of glucose that plants produce in photosynthesis. So what plants will then do is combine 2 of these to make a 6-carbon carbohydrate, such as glucose. So glucose has the 6 carbons, so it has the formula C6H12O6. So again, just twice as many hydrogens as there are carbons and oxygens. But there's another thing here that you might have noticed, and that's that we also get two other monosaccharides that have the same molecular formula. So fructose and galactose. So fructose and galactose also have the formula C6H12O6. So how is that possible? Well, if you look at the diagrams at the bottom there, what you can see is that they have different structures. So they can have the same molecular formula because they're made of the same atoms, the same building blocks. So each of them has 6 carbons and 12 hydrogens and 6 oxygens. They're just arranged slightly differently. So you can think of it as if I were to give you 6 yellow Lego bricks and 12 red and 6 blue, you could arrange those Lego bricks in a range of different things. You could build a car and a house and a tree. And it's the same principle here. We have the same building blocks. They're just put together in a slightly different way. And we call these structural isomers. So structural isomers then are where we have the same molecular formula, but they're just arranged slightly differently, so we get different structures. And what you'll also notice in the diagrams there is that we have these ring structures with our carbohydrates. And this typically happens when carbohydrates are put into water. They'll actually bend around to form these ring structures. And when they actually fold around or bend around to make the rings, the join is typically represented over here on the right hand side. So we typically will when we're referring to the molecules, we'll start over here with this carbon. So this would be carbon number 1. And over here on fructose, we would say that this is carbon number 1. And over here on galactose, we would say that this is carbon number 1. One other point that is worth mentioning now before we move on is if we look at glucose here, and we can see its structure, and here we can see the 6 carbons and 12 hydrogens and 6 oxygens. But glucose itself actually comes in 2 different forms, and they are called alpha and beta glucose. And you might hear them mentioned. I don't really refer to them that much, but just in case you do hear them referred to, it's all about this first carbon and the attachment of this oxygen and hydrogen down here. And they're in a form of what we refer to as a hydroxyl group. And then up here, we've got this lone hydrogen. So when they're in this configuration with the hydroxyl group at the bottom and then the hydrogen at the top, we'd refer to this as an alpha glucose. If these two were reversed, so the OH or the hydroxyl group was at the top and the hydrogen was at the bottom, that would be called a beta glucose or beta glucose. And this creates differences in how the glucoses will join together with other glucoses, and then we can get different structures. So for instance, alpha glucoses can join together to form starch, and then beta glucoses tend to join together to form cellulose. So that will become important in a bit. I've mentioned most of these, or 3 of these so far, but I just wanted to show you the 5 main monosaccharides that we'll be dealing with in our sessions together. And we can see here that, again, recap, monosaccharides refer to single versions of each sugar. And these 5 common monosaccharides are glucose. So you quickly answer to yourself, is this an alpha or a beta glucose based on the arrangement of these atoms, or the hydroxyl group and the hydrogen of this first carbon? Yep, it's an alpha glucose. And then we've got fructose, and then we have galactose over here. Have you noticed that these are often referred to as hexoses? So hex referring to 6. So these are 6 carbon sugars. Down here, we've got a couple of pentoses, so pent referring to 5. So these are 5 carbon sugars. So they would have the formula C5H10O5. Apart from deoxyribose, actually, which is missing an oxygen, but we'll talk about that in a later lesson. But these 2 will become very important in the session where we look at nucleic acids, because we find deoxyribose in DNA. It's a very important sugar in forming the monomers, which are called nucleotides, that will make up our nucleic acid of DNA. And then we get ribose, which is very important in the nucleic acid called RNA. So we'll see these 2 in the next session. But for now, those are the 5 main monosaccharides that we'll be referring to. And then we can join 2 monosaccharides together to get a disaccharide then. So these are 3 of the most common disaccharides that we'll be referring to. At the top here, we can get sucrose. So sucrose is just normal table sugar, and we tend to get that from sugar beet. And sucrose is made up of a glucose and a fructose. So you can see the glucose and then the fructose. And you can see the bond between them is through this oxygen here. And we're going to go on to look at where this comes from in a minute, how this bond is made. Next one down is one that you've probably all heard of before, and that's lactose. And that's made up of a galactose and a glucose. So we can see those 2 here. And again, we can see the bond here. So I'll mention it now, but I'll talk about it again in a bit. When we get this bond or this link between 2 carbohydrates like this, it's called a glycosidic link. And again, I'll talk to you about that in a bit more detail in a bit. And then down here, if you enjoy beer, you'll be familiar with this one. This is maltose, so it's just made up of 2 glucoses. So this is the sugar that gives beer its sweet taste. So all praise be to maltose. So those are a few examples of disaccharides then, so double sugars. And then we go on to the polysaccharides, so many sugars. And these are made up of hundreds or thousands of monosaccharides joined together. And in this case, these are these 4 examples are all made up of glucoses all joined together. So what we've got here are 2 examples from plants and 2 examples from animals. And we've got examples of structural polysaccharides. So those are polysaccharides that produce physical structures to provide strength and rigidity. And then we also have a couple examples of storage polysaccharides. So when an organism has a surplus of the monosaccharides, it has a surplus of carbohydrates, it can actually bind them or bond them together and store them for later use. So right at the top here, we have cellulose, and cellulose is made up of lots and lots and lots of glucoses, beta glucoses, all joined together. So you can see the bonds here, and here, and here. And then this is a structural polysaccharide. So it makes up the cell walls of every plant cell, whether it's single-celled algae or whether it's an oak tree. All of the plant cells will be made up of cellulose. So it's a really, really important polysaccharide. But plants can also store carbohydrates in the form of starch. And you can see starch here. So we're already seeing an example of 4 of the monomers joined together, but this will be sort of hundreds or thousands of glucoses joined together. So if you think about you can think about the situation, or that you can picture it. So if you're a potato plant and you're happily growing away, well, when spring and summer come about, suddenly you have access to longer day lengths and brighter and stronger sunlight. So you can spend your days busily photosynthesizing and producing lots of sugars. In fact, you produce way more than you need. So instead of sort of wasting that, you can send it down to your roots and you can turn it into starch and you can store it. And that's what we see as potatoes. So that potato is a store of starch that the plant has produced for using later on, or for growing an entirely new plant. Obviously, we can benefit from that by eating it and breaking down that starch and accessing the glucoses that make it up. And we'll talk about how we do that in a bit. So both of these are found in plants then. Cellulose is a structural polysaccharide. Starch is a storage polysaccharide. But here are 2 more that we find in animals. So glycogen is a storage polysaccharide found in animals. So when we have an excess amount of carbohydrates in our diet, what we can actually do is convert that into glycogen. We can store these glucoses, we can join them together through dehydration synthesis, and we'll actually store them in our liver and in our muscles. And what that then will act as a really easy easily accessible store of glucose. So when our blood glucose levels drop, we can actually produce, or we do produce, a hormone called glucagon that can actually break that glycogen down then that's in our liver and our muscles, turn it into glucose that we can then use to turn it into energy. If we have, once our glycogen stores are full, we can actually then convert any additional carbohydrates into fat. And fat is a much more efficient way of storing energy for storing fuel long term. So a gram of fat will have twice as much energy in it as a gram of glycogen, for instance. But it's harder for the body to access that fat and convert it back into carbohydrates, for instance. Whereas glycogen is very, very easy to break down. So we store it in the liver and the muscles, and then we can easily access it. So that's a storage polysaccharide found in animals. The bottom one is called chitin, and that is a structural polysaccharide found in animals, but also it's found in fungi. So it makes up the cell walls of fungi, so mushrooms and toadstools and things like that. But it's also a really important component in the exoskeletons of arthropods, so crabs, lobsters, insects, things like that. And you can see there that it's glucose, but it also has the addition of some nitrogen in it as well. So it's a slightly different structure. But there we can see then two examples from plants, cellulose and starch, and then two examples from animals, storage polysaccharide in the form of glycogen, and then a structural polysaccharide in the form of chitin. What we do now is we can pause the video and then just jot down the answers to these questions then. And then once you've done that, we can resume and I'll go through the answers with you to these little recap questions before we move on to the next section. Okay, so number one, name 3 monosaccharides. And then number 2, name 2 disaccharides. Number 3, name 2 storage polysaccharides. And then number 4, name 2 structural polysaccharides. So we'll give it a minute. You can answer the questions and then I'll go through the answers. Okay, so welcome back. Let's go through the answers then. Hopefully they were all fresh in your mind and you didn't have to revert back to your notes. But don't worry if you did. I won't tell anyone. So, question number 1 then. Name 3 monosaccharides. Well, I gave you 5 examples. So hopefully you were able to name 3 from those. So you could have had glucose, fructose, galactose, ribose, or deoxyribose. So 3 of those. Well done if you got all 5. So number 2, name 2 disaccharides. Well, we looked at 3 examples. So you can have 2 of these 3. So sucrose, lactose, or maltose. Again, well done if you got all 3. And then question number 3, name 2 storage polysaccharides. Well, we just did those, so hopefully they're fresh in your mind. Starch in plants and then glycogen in animals. And then number 4, name 2 structural polysaccharides, and they are cellulose and chitin. Okay, so don't worry if you didn't get all of those right. It's good to keep testing yourself and questioning yourself. You can go on to Google actually and type in carbohydrates quiz, and it will bring up lots and lots of additional sort of multiple choice questions and things like that. Testing yourself is a really, really good way to see how you're doing and highlighting the areas in which you need to improve or go back over. Okay, so now we've had a look at what carbohydrates are, and we've talked a little bit about where they come from, and we've had a little look at their structure. Next thing we need to do is look at the process of making and breaking the large polysaccharides and even the disaccharides. Because the problem that we have is when we consume our food, so say for instance you were to have some chips for your dinner, the starch in the potato is a really big molecule, and it's far too big to get from your digestive system into your blood and then into the cells where it's needed. So the first process that the body needs to do, and this is all part of the digestive system, is to actually break down those macromolecules, those big carbohydrates, but also proteins as well, and nucleic acids. We need to break those down first, so they're back to their monomers, so they're small enough to get into our blood and then delivered to our cells. Once inside the cells, certain molecules can then be built back up into larger molecules, or in the case of something like glucose, it can get broken down and used for the production of energy. The process of breaking our macromolecules is called hydrolysis, and the process of making them in the first place is called dehydration synthesis. So I've got a short animation here to show you what that process looks like. So first we're going to start off by combining or joining two monomers, and then we'll look at the through dehydration, and then we'll look at the process of hydrolysis where we break them down. And then we'll go through and talk a little bit more about them. So we'll have a look at this animation.
Most biological molecules are very large and are built by assembling small molecules, or monomers, into long chains. The resulting molecules are called macromolecules, or polymers. A process of linking monomers, called dehydration synthesis, involves the removal of 2 hydrogen atoms and 1 oxygen atom to form water. One way this might happen is diagrammed here, where several generic monomers are shown with OH groups that could be used for linking. The animation illustrates the joining together of monomers by dehydration synthesis, catalyzed by a polymerase enzyme, and the reverse process, in which added water results in hydrolysis, catalyzed by a hydrolase enzyme.
Hopefully that was easy enough to follow. But important things that you could see there was this process, or the processes, both making macromolecules through dehydration and then also breaking them apart through the process of hydrolysis. Both of those require the help of enzymes, and enzymes are a really important group of macromolecule. Typically, they're proteins, and I've got a whole lesson on those that we'll cover in a few weeks' time. But we also saw that water was heavily involved in this process as well. So as we go through, we'll talk a little bit more about that. Before we get into looking at dehydration and hydrolysis in more detail, we need to first, I wanted to introduce you to these things, which are called functional groups. So as we can see here, functional groups are groups of atoms that have predictable behavior and are typically the part of a molecule that takes part in chemical reactions. So in biology, in biochemistry, there are 7 functional groups that we encounter. But for the moment, I just want to introduce you to these 4. So these 4 functional groups are ones that we're going to encounter over the next few sessions. So I just wanted to highlight them at this point so you can look out for them as we go through. We'll start down here on the bottom left because this is the one that's going to be most important to us today. And this is called a hydroxyl group. And it's just an oxygen covalently bonded to a hydrogen. And we typically just write it as an OH, so an oxygen and a hydrogen next to each other without the line. If you remember from the last session, we know that oxygen, with its atomic number of 8, has 8 electrons, so 2 in the first shell and then 6 in the second shell. Oxygen wants to make 2 more bonds to get hold of those 2 additional electrons to complete its valence shell. So 1 bond goes to the hydrogen, so that's a covalent bond there, but it also means then that oxygen can attach to something else. So we'll see these hydroxyl groups heavily important, or very important, in our carbohydrates and also in the making and breaking of bonds between carbohydrates. Another one that we see up here is called a carbonyl group, up in the top left, and that's a carbon double bonded to an oxygen. And as we know from last week, carbon, with its atomic number of 6, has 4 valence electrons, so it can make 4 additional bonds. 2 go into the double bond with the oxygen, and that means that carbon can make 2 additional bonds. So we'll see these carbonyl groups in the next lesson. If we combine the hydroxyl group and a carbonyl group, so you can imagine we can take this hydroxyl group here and we attach it here to this carbonyl group, so you could imagine coming down here, you then have the OH, you get this functional group, which is a carboxylic acid group, sometimes just shortened to a carboxyl group. But its full name is the carboxylic acid group. And there's the carbonyl group, carbon double bonded to an oxygen, and there's the hydroxyl group there. The fourth one that we're going to look at, and we'll look at this one next week, is an amine group or an amino group. And that's just a nitrogen covalently bonded to two hydrogens. Nitrogen wants to make 3 additional bonds, so it can attach to something else here. So the reason I wanted to highlight these now is when you see these as part of a molecule, it tends to tell us that this is where, this is the part of the molecule where a chemical reaction is likely to take place. So as we go through, we can highlight particularly the hydroxyl groups. Making macromolecules then. So we know that this comes through or is done through a process called dehydration synthesis. So monomers are linked by covalent bonds and through the loss of a water molecule in dehydration synthesis. So a couple of important points there is that what we are creating here are covalent bonds. So we know from last week or from the last session that this is the sharing of electrons. And each time we create one of these bonds through dehydration synthesis, a water molecule, so H2O, is given off or is released. And that's why this is called a dehydration or a condensation reaction. It's because water is removed during the process. And remember that this requires the help of an enzyme to do it. The process is facilitated by specific enzymes, and we'll talk about those enzymes in a couple of lessons' time. Looking at this example then, we can lead to fairly simple molecules such as maltose, but we can also make very, very large macromolecules such as starch and cellulose. But on the diagram here, we've got the example of maltose. So we know that maltose is made up of 2 glucoses joined together. So here's one glucose, and have a look at what we have here. I mentioned it just now and also earlier on. This is a hydroxyl group. It's one of our functional groups. And if we look at the next glucose, we can see that right here is another hydroxyl group, so another OH. And what will happen is an enzyme will come along and it will sort of attach itself to these 2 glucoses. And enzymes catalyze chemical reactions. They speed up and facilitate these chemical reactions. So what the enzyme is going to do is it will actually remove a hydroxyl group from one of the glucoses, and then it will remove a hydrogen off the other one. And then 2 hydrogens and an oxygen make our water molecule, our H2O. The enzyme will then facilitate the forming of a covalent bond between this oxygen and this other glucose. So now, if you remember, I said that where we get these bends, these represent the carbons. So these 2 carbons will now be joined between that oxygen. So if we look over here, we can see maltose. This is the carbon of the first glucose covalently bonded to the oxygen, and this is the second glucose covalently bonded to the oxygen. And this is now joined together. This is now the disaccharide maltose, and this link between the 2 of them, formed by dehydration synthesis, is called a glycosidic link. I mentioned it earlier, and you can see it there. A glycosidic link is that link between 2 carbohydrates. So what we can obviously do now is we can keep adding more and more glucoses onto here until ultimately we can end up with our polysaccharides like starch and glycogen and cellulose and chitin. They're all made through the same process, through forming glycosidic links through dehydration synthesis. Each time you form a link, you need an enzyme to do it, and each time a link is formed, you do so by removing a molecule of water. And that's it. That's the process of dehydration synthesis. This is how this is how we make complex carbohydrates. This is also going to be how we make proteins, and this is also how we make big molecules such as nucleic acids, which are DNA and RNA. So please take the time to go over this after this session, just to get it comfortable in your head, because we are going to see this process quite a bit over the next few sessions. So the way I always think of it is dehydration builds, and as we'll see in a minute, hydrolysis breaks. So dehydration builds our molecules, hydrolysis breaks them apart. So speaking of hydrolysis then, we can see here that polymers are disassembled to monomers by hydrolysis. So hydro referring to water, lysis means to break, so hydrolysis, to break water. And hydrolysis is essentially the reverse of a dehydration reaction. So we see there, hydrolysis means to break water, and it breaks the bonds between monomers by the addition of water. So when with dehydration, we actually removed a molecule of water, with hydrolysis, we're going to add it back to where it was originally. If we look at the diagram down here, we can see that we have the disaccharide sucrose, so glucose on the left, fructose on the right. There's that glycosidic link joining them together. Well, plants make sucrose because it's a more stable molecule for transporting glucose and fructose from their leaves, where it's produced during photosynthesis, down to their roots, where they can then store it. So sucrose is a much more stable molecule. So they will actually form this by taking a glucose and a fructose, and then through dehydration synthesis, will then join them together to then transport those sugars around their bodies. Obviously, then we come along and we consume the sucrose, but again, this is too big to get into our bloodstream, so we have to first break them apart into their monomers, into the glucose and fructose. We do that through the help of an enzyme again, but we also need a molecule of water. So H2O comes along, the enzyme will break the glycosidic link, and then it will take one of the hydrogens from H2O and it will add it to the oxygen, which is attached to the, in this case, into the fructose, and then the other hydrogen and the oxygen from the water molecule forms our hydroxyl group, which is then covalently bonded to the carbon of the glucose. So that effectively breaks that glycosidic link and then gives us our glucose molecule and our fructose molecule, and that is now an ideal situation because those molecules are small enough to get into our bloodstream, so we can get them into our cells where we can use them. So that is simply how hydrolysis works. And again, it doesn't matter whether we're just talking about breaking a simple carbohydrate like sucrose into two, or whether we're breaking down a much larger molecule like glycogen or starch. It's the same process. Every time you want to break off one of these carbohydrates, every time you want to break a glycosidic link, you need an enzyme to help you, and you need a molecule of water to re-establish the hydroxyl groups between the monomers. So the classic example of hydrolysis working in our bodies then is digestion. Every time we consume carbohydrates or proteins, we have to break those big molecules down into their monomers, and we do it through hydrolysis. And this just confirms what I've already said. So the majority of organic material in our food is far too large to enter our cells. So it's broken down to monomers by enzymes in the digestive tract. And then once it's inside the cells, it can be reassembled into whatever the cell needs. So those are the processes of dehydration synthesis and hydrolysis. And I'll revisit these again in the next session because they are very important. Okay, so let's do another little recap session then. What are the four main classes of large biological molecules? So hopefully you remembered that they are carbohydrates, lipids, proteins, and nucleic acids. Number two, what happens to water during hydrolysis? So quickly write that down. So what happens to water during hydrolysis? Slightly trickier to put into words, this one. Okay, so I'll give you the answer now. So it's consumed, or it's used up, it's broken apart. any sort of form of that would be acceptable in your description of hydrolysis. So to break a bond, you use hydrolysis, and hydrolysis means to break water. So what happens to water during dehydration synthesis then? So again, jot down your answer as best you can. Don't worry too much about using the right words at this stage. Just try and get the main concept down. Okay, so let's go through the answer then. So it's produced, or it's given off, or it's removed, or some form of that is what I was looking for at this point then. So every time a new bond is formed during dehydration synthesis, so in the case of carbohydrates, it's when we produce a glycosidic link. Every time we produce a link, a molecule of water is produced, or it's removed. Okay, so you can test yourself on this then now. So number 4, how many molecules of water are needed to completely hydrolyze a polymer that is 10 monomers long? So if you imagine you've got if you remember, we said that the carbohydrates, with carbohydrates, those that are made up of 3 to 10 monomers are called oligosaccharides. So imagine you have an oligosaccharide made up of 10 individual monomers, and you want to break those apart. How many molecules of water would you need? It can help if you just quickly draw it out. Let's give you a minute to answer that. Okay, so let's quickly go through the answer then. So how many molecules of water are needed to completely hydrolyze a polymer that is 10 monomers long? Well, the answer is 9. So with 1 water molecule required to hydrolyze each connected pair of monomers. So a simple way of doing that is if you imagine that each monomer is a circle, and you could just draw 10 circles right next to each other. Each join between the circles would represent a glycosidic link or a bond. So there will be 9 of those. So you'd need a molecule of water to break each one. Okay, so that brings us to the end of our introduction to organic compounds then. So we looked at our first class, which is carbohydrates. So we've talked a little bit about carbohydrates. So hopefully now you would be able to identify examples of monosaccharides, of disaccharides, and then also polysaccharides, including hopefully you'd be able to identify the difference between structural and storage polysaccharides and give examples of where we find them. And then also we talked about dehydration synthesis and hydrolysis. So remember, dehydration builds and hydrolysis breaks. So if this is the first time that you're learning this, then it can be a lot to take in, but sort of go back over it a few times, try and answer some questions. Like I said, you can go onto Google and type in carbohydrates quiz, and it will bring up probably an infinite amount of them. So you can go through some and just practice and practice. And I think the concepts that we've covered here are fairly straightforward. The difficulty comes in the terminology. It's remembering all of those words. So try and find a way that works really well for you in terms of learning this. So it might be that you work well from doing quizzes, like I just said, or it might be that just writing down bullet points of what you remember. So you can put monosaccharides and then just bullet point what you know about monosaccharides. So they're single sugars, they're simple sugars. They have a formula of CH2O. You've got examples such as glucose and fructose and galactose. And so you can do it that way. or you can sort of mind map it or do a spider diagram where you put disaccharides in the middle and then write everything that you know about disaccharides. So try a few different versions of this, try different methods and see what works for you. And what you'll find is hopefully you'll stumble on a process that really clicks with you and helps you to learn these topics, because like I said, most of the time the processes are fairly straightforward. It's just getting used to the terminology and what all of the words mean and how to use them in the right way. But anyway, hopefully that was helpful. In the next session, we'll have a look at lipids and nucleic acids, and then ultimately going on to proteins to complete our look at the 4 classes of organic compounds. Okay, thanks very much.